Plants were winning evolutionary battles millions of years before humans even existed. And the weapons they used? Invisible chemistry so sophisticated that it still baffles modern science. These compounds, buried in bark, suspended in resin, scattered across petals, they were never meant for us. And yet, we discovered they could heal us. Inspire us, you found dream and cognition. A space where the deepest questions about your mind, your sleep, and the natural world finally make sense. Let's begin. Even prepare our minds for sleep. Tonight, we're unwrapping one of the most underestimated stories in human history. How a species learned to read the chemical language of another species and use it to transform civilization. From the spices that funded empires to the essential oils that quiet your mind at night, every single one has a story written in molecular detail. A story of defense, of seduction, of pure survival. And as you drift deeper into this exploration, you'll begin to see the world differently, recognizing that the gentlest sensations, the most calming moments, are all rooted in chemistry so elegant it almost feels like magic. So relax, let your defenses lower, because understanding this hidden world might just change how you experience rest forever. Let's begin by understanding something remarkable about plant extracts. They can heal, poison, perfume, and preserve all at once. Across the span of human history, people learned that the green world around them held extraordinary gifts. The same plants that could soothe a fever could also stop a heart. The same substances that could fill a room with fragrance could keep decay away. This is what makes plant extracts so astonishing. They are not single-purpose inventions designed with one narrow task in mind. Instead, they are compact chemical toolkits, each one equipped with multiple powers waiting to be discovered and understood. A resin from a tree might seal a wound and simultaneously slow the growth of harmful microbes. A flower oil might lure pollinators in the wild, then centuries later become part of a palace perfume, serving an entirely different purpose. A bitter alkaloid might defend a leaf from hungry insects in the forest, then become a medicine in the right dose, or a poison in the wrong one. Even the spices sitting in our kitchen cupboards often carry protective chemistry that once helped the plant survive intense heat, creeping mold, or animals looking for a meal. When humans first learned to draw out these hidden substances, they were not creating magic from nothing. Rather, they were opening a cabinet of powers that plants had been quietly building for millions of years, refining their chemistry through countless generations. A single leaf can hide hundreds of powerful chemical compounds working in perfect concert. When you look at a leaf from a distance, it appears simple, flat, green, silent, almost ordinary. Yet inside that small piece of living tissue lies a whole laboratory at work. Plants do not create just one useful substance. They produce families of compounds, each with its own purpose and role. Some compounds protect the plant against fungi. Others repel insects that would otherwise consume it. Some absorb sunlight safely and convert it to energy. Still others repair damage caused by heat or drought stress. Beyond survival, plants craft molecules that shape scent, taste, and color, the very qualities that make them attractive to pollinators and dispersers of their seeds. Scientists who analyze a single leaf discover an intricate crowd of molecules, each with a distinct role, each produced at precisely the right place and moment in the plant's tissue. Mint leaves carry cooling compounds that create that distinctive sensation on the tongue and skin. Tea leaves hold bitter tannins, caffeine, and aromatic molecules that shift and transform during processing, revealing different flavors and effects. Even something as simple as crushing a tomato leaf releases a sharp green smell, a signal created when damaged cells suddenly mix chemicals that had been stored separately, their encounter triggering an immediate response. So when you hold one leaf in your hand, you are not holding a single ingredient. You are holding a crowded chemical city, a metropolis of purpose and function, hidden in plain sight. Humans have built entire systems of medicine by studying what plants already make. Long before laboratories and microscopes existed, people watched the living world closely. They noticed which roots eased pain. They learned which barks lowered fevers. They discovered which seeds were too dangerous to touch. Much of medicine as we know it began there, with human beings learning from plant chemistry step by step, through careful observation and accumulated wisdom. Willow bark pointed the way toward the familiar pain relief we use today. Foxglove revealed compounds that could slow and steady a struggling heart, transforming the treatment of cardiac conditions. Cinchona bark offered quinine, which became one of the great anti-malarial discoveries in history, saving countless lives across continents and centuries. Later, chemists learned how to isolate these substances, refine them, test them in controlled ways, and sometimes reshape them into safer or more reliable drugs. Yet the starting point was always the same. Nature had already done the hard part. Plants had already solved the chemical problems that humans were struggling with. We simply had to learn to listen. So as you settle deeper into this moment, remember that the world around us is filled with hidden chemistry. Silent, patient, and infinitely complex. The next time you hold a leaf, smell a flower, or taste a spice, you're touching millions of years of botanical evolution, a library of solutions that nature has been writing all along. Sleep well tonight knowing that this green wisdom surrounds us, waiting to be understood and appreciated. The plant had evolved a molecule with powerful effects, and humans learned how to listen. That is one of the reasons plant extracts have remained so compelling across every era of history. They are not relics from a distant past. They are clues and sometimes the first whisper of tomorrow's medicine. Each discovery is a reminder that nature has been conducting its own experiments long before we ever thought to begin ours. Vanilla's most famous flavor begins deep inside an orchid seed pod, and its story is anything but ordinary. Vanilla feels familiar to us now, present in cakes, ice cream, candles, and perfumes. Yet it begins its life in a climbing tropical vine whose delicate flowers open only briefly. If pollination succeeds, the plant slowly grows long green pods packed with tiny seeds and the raw materials of future aroma. But here is the surprising part. Fresh from the vine, those pods carry none of the scent most people associate with vanilla. That familiar richness does not simply exist, waiting to be plucked. It has to be coaxed into being. The transformation happens through careful curing, through heat, drying, and time. These conditions allow enzymes and stored compounds inside the pod to interact and change, slowly unlocking that deep, sweet warmth we recognize. It is one of the most labor-intensive flavor journeys on Earth, hidden inside the fruit of an orchid most people would never recognize if they passed it in the wild. Caffeine is a plant defense that humans transformed into a daily ritual. To us, caffeine is morning itself, the first sip of coffee, the warmth of tea, the small and gentle lift that helps the world come into focus. To the plant, it began as something entirely different. Caffeine is defensive chemistry, produced in leaves and seeds to discourage insects and interfere with the bodies of creatures small enough to be harmed by it. Some plants even release it into the soil, where it may suppress the growth of neighboring competitors. Yet when humans encountered this bitter stimulant, they did something plants could never have planned for. We made it social. We built ceremonies around it. Coffee houses became places of conversation, debate, and commerce. Tea became woven into traditions of hospitality, discipline, and quiet pause. Across continents and cultures, the same plant compound journeyed from evolutionary defense into the heart of human culture. That is part of its wonder. A molecule shaped by survival struggle ended up embedded in breakfast tables, night shifts, study sessions, and shared afternoons, quietly guiding the rhythm of modern life. Some of history's most feared poisons began as ordinary plant extracts. Many deadly plant toxins do not come from exotic creatures lurking in remote jungles. They come from leaves, seeds, roots, and flowers that can look almost harmless. And that is part of what makes them so unsettling. Because plants cannot run from danger, some evolved chemistry powerful enough to stop an animal after a single bite. Over time, humans learned to recognize these hidden dangers and, in some cases, to exploit them. Deadly nightshade carries compounds that can disrupt vision, heartbeat, and awareness. Hemlock played a role in one of history's most famous executions. Castor beans contain resin, one of the most feared natural toxins known to science. Even ornamental plants growing quietly in gardens can hold potent cardiac or nerve-affecting chemicals. None of this is accidental. It is survival written in molecules. A plant with strong chemical defenses is far less likely to be eaten, stripped, or destroyed. Behind the beauty of flowers and foliage lies an ancient truth. nature's chemistry can be elegant and merciless in equal measure. The scent of a rose comes not from a single note but from a chemical orchestra playing in perfect concert. When someone says a rose smells like a rose, it sounds simple, but the fragrance itself is anything but. Rose scent is built from dozens of aromatic compounds rising together in shifting proportions, and that blend can differ from one variety to the next. Some molecules bring sweetness, others add spice, fruit, citrus, or a faint green freshness underneath. What reaches your nose as one seamless, effortless perfume is really a layered performance, with each chemical adding depth, brightness, softness, or lift to the whole. Temperature matters. Time of day matters. Even the stage of the bloom matters. A rose opening in cool morning air may smell distinctly different by the warmth of afternoon. This complexity is why rose perfume has fascinated growers, distillers, and chemists for centuries. They are not chasing a single ingredient. They are trying to capture an accord so perfectly balanced that the mind reads it as pure, effortless beauty. Behind that familiar floral breath is a remarkable act of natural composition, as precise in its own way as music itself. Some of the most powerful effects in daily life arrive in vanishingly small amounts. The trace compounds in a spice, the microscopic droplets of essential oil released by a crushed herb, the faint molecular signals drifting from a flower, these are not small in their impact. They can transform food, shift mood, and unlock memories so vivid they feel almost physical. The chemistry of plants operates at scales our senses were never designed to measure. Yet somehow, without any instruction, we have always known how to feel it. A few drops of vanilla can transform a batter from flat and forgettable into something warm and inviting. A trace of mint can make water taste colder than it actually is. The scent of orange peel or crushed basil can shift the feeling of an entire room before anyone has said a single word. Plant extracts work this way because our senses are exquisitely sensitive to certain molecules. They do not need much. A little can awaken appetite, comfort, nostalgia, alertness, or calm. Smell is especially powerful in this regard, because it is closely tied to memory in ways that no other sense quite matches. One fragrance can carry a person instantly back to a childhood kitchen, a garden after rain, or a holiday table long since gone. That is why plant chemistry feels so much larger than its size. Tiny quantities can reshape experience itself, changing what food tastes like, how a space feels, and which forgotten corners of life suddenly come rushing back. Many plant extracts evolved first as weapons in nature's silent and unending wars. Plants may appear passive from a distance, but they live in constant conflict. They are chewed, pierced, infected, shaded, crowded, and attacked from every side. Because they cannot flee, many evolved chemistry as their primary form of warfare. Bitter alkaloids discourage herbivores. Sharp sulfur compounds burst into action the moment tissues are damaged. Sticky resins trap insects and seal wounds. Some roots release substances into the soil that make life harder for nearby rivals. Other plants send airborne chemical signals after an attack, warning neighboring plants that danger is close. Seen in this light, a forest or meadow is not chemically peaceful at all. It is full of defenses, traps, alarms, and countermeasures moving silently through air, sap, bark, and leaf. This is where so many plant extracts truly began, not as flavorings, remedies, or perfumes for human use, but as survival tools forged under millions of years of evolutionary pressure. We entered the story much later, discovering that nature's weapons could also become our medicine, our spice, and our scent. Mint feels cold because its extract quietly tricks your nerves into believing so. When mint touches your tongue or skin, nothing actually becomes colder. Instead, menthol binds to receptors that normally respond to a genuine drop in temperature, and your nervous system reports coolness even when the air or liquid around you remains exactly the same. That is why mint can make chewing gum feel icy, toothpaste feel fresh, and a breeze across mentholated skin feel suddenly, unmistakably sharper. It is a chemical illusion, but a remarkably convincing one. Plants did not evolve this sensation to brighten human breath. Mint most likely uses menthol and related compounds as protection, making itself less appealing to insects and animals that might otherwise consume it. Yet to us, the effect feels clean, vivid, and strangely invigorating. One small leaf can therefore rewrite your sensory world for a moment. It can persuade your body that cold has arrived out of nowhere, proving that plant extracts do not simply flavor experience. Sometimes they edit perception itself right at the level of the nerves. Chilly heat comes from a plant compound that mammals have learned, through long experience, to fear. The fire in a chili is not a taste in the ordinary sense. It is pain. Capsaicin binds to the same receptors that respond to genuinely dangerous heat, so your mouth reacts as though it has touched something burning. Your heart may quicken, your eyes may water, your skin may flush. For the plant, this was a remarkably clever evolutionary move. Mammals tend to crush seeds with their teeth, destroying the plant's future. Birds, however, do not sense capsaicin the same way, so they can swallow the fruit whole and carry the seeds elsewhere. In other words, the pepper found a way to discourage the wrong visitors and welcome the right ones. Then humans complicated the story entirely. We turned that warning signal into cuisine, competition, and craving. We chase the burn, sweat through it, and come back for more. Few plant extracts show human nature more vividly than the chili, a compound that began as a deterrent and became a source of deep, almost defiant delight. Lavender extract influences insects long before any human being stops to smell it. Before lavender became associated with linen, soap, and evening calm, its fragrance was already shaping the lives of much smaller creatures. The oils rising from its flowers are signals in the insect world, some guiding pollinators toward nectar, others discouraging pests or interfering with insects that might otherwise feed on the plant. To a bee, the scent is not a decorative extra. It is part of a map. To a hungry herbivore, it can be a warning. What humans notice as softness or freshness is therefore only one layer of a much older biological language. Lavender is broadcasting constantly, and insects are among the first to hear it. A calm-smelling bloom standing quietly in a garden is also an active chemical beacon, negotiating with pollinators, warding off trouble, and managing its place in the landscape. Its perfume reaches us last, after evolution has already assigned it work far older and more urgent than relaxation. Nicotine is a plant insecticide that crossed into the human brain and never left. Tobacco did not evolve nicotine for human fascination. It evolved it as a weapon. In insects, nicotine attacks the nervous system with brutal efficiency, overwhelming the signaling pathways that living bodies depend on to move and survive. That made it a highly effective defense in the ancient contest between leaf and predator. Humans discovered the same molecule and found that in smaller doses it could sharpen attention, alter mood, and create a rapid feeling of reward. The reason lies in the brain, where nicotine can mimic one of the body's own signaling chemicals and trigger pathways linked to pleasure, learning, and repetition. That is how a plant defense became one of history's most powerful human habits. It crossed from field to culture, from ecology to addiction, carrying both stimulation and danger within the same compact molecular structure. Few extracts demonstrate more clearly how a single molecule can be useful, seductive, and destructive all at once, without ever changing its fundamental nature. Quinine from bark once helped humanity push back against one of its oldest and most relentless enemies. There was a time when vast parts of the world were haunted by a fever that seemed almost inescapable. Malaria could hollow out armies, stall entire trading routes, and make whole landscapes deadly to enter. Then came the bark of the cinchona tree, carried down from the Andes and prized for the bitter compound hidden within it. Quinine did not end malaria, but it gave people a powerful tool against one of history's great killers, and that changed exploration, medicine, empire, and survival itself. Travelers, soldiers, and doctors all came to depend on a substance drawn from a tree that had evolved its chemistry for entirely its own reasons. The taste was harsh, but the effect could be life-saving. Even tonic water today carries a faint echo of that story. Behind its mild bitterness lies a chapter in which plant extraction helped humans push back against disease and move through regions that had once seemed nearly impossible to endure. Foxglove compounds changed the course of heart medicine by revealing that a failing pulse could sometimes be steadied by chemistry hidden inside a garden flower. Foxglove is one of those plants that seems almost theatrical. Tall spikes of purple flowers rising from the ground with a beauty that invites quiet admiration. Yet inside the plant are compounds powerful enough to alter the rhythm of the human heart. In carefully controlled doses, substances derived from foxglove help doctors strengthen weak contractions and slow certain dangerously rapid heartbeats. That was a profound discovery. It showed that medicine need not always arrive from a laboratory bench. Sometimes it was already growing in the garden, waiting to be understood. The margin for error, however, is narrow. Too much can become dangerous very quickly, a reminder that even the gentlest-looking plant, foxglove, holds a special place in medical history, poised precisely between remedy and risk. It showed that the line between healing and harm is often measured not in different substances, but in different amounts. Few examples capture the drama of plant extracts more vividly than this. A blossom that looks almost storybook gentle, capable of reaching directly into the machinery of life itself. Grapefruit arrives with bright color, fresh scent, and the promise of sweetness. Yet the first bite often carries a surprise. Beneath the juicy brightness lies an assertive bitterness that sets it apart from most other citrus fruits. That character comes from compounds such as naringin, which give grapefruit its unmistakable edge and make it feel more like a deliberate awakening of the palate than a simple pleasure. But grapefruit chemistry is fascinating for another reason entirely. Its extracts can interact with enzymes in the body that help process certain medicines, sometimes changing how strongly those medicines act. So this everyday breakfast fruit is not chemically simple at all. It is a vivid reminder that flavor and physiology can meet in unexpected ways. A pink wedge on a plate can carry beauty, bitterness, and biological consequence all at once, turning an ordinary kitchen moment into something far more chemically dramatic than it appears. Tea leaves hold entirely different extracts depending on how they are processed, and that difference is profound. Black tea, green tea, white tea, and oolong can taste so distinct from one another that they seem to come from separate plants entirely. Yet many begin as leaves from the same species. What changes the experience is what happens after picking. The leaves may be heated quickly to halt certain chemical reactions, or they may be allowed to oxidize, curl, darken, and slowly transform. During that journey, the balance of compounds shifts in remarkable ways. Fresh grassy notes may soften, floral hints may deepen, tannins may become rounder or more pronounced. Entirely new aromatic molecules can appear as the leaf's chemistry rearranges itself after harvest. This is what makes tea so endlessly enchanting. It is not simply a plant ingredient dropped into hot water. It is a record of choices made after picking, each one shaping what the cup will ultimately become. The same hillside leaf can end as something brisk, smoky, buttery, floral, malty, or deep, depending entirely on how human hands choose to guide its chemistry. Coffee aroma does not exist in the raw bean. The famous scent that draws people in each morning is born entirely in heat. Before roasting, a coffee bean holds little of the magic people line up for. But as the beans roast, sugars, acids, proteins, and stored compounds begin to react in waves, generating a dense cloud of entirely new molecules. Some smell nutty, some smell of caramel, fruit, smoke, chocolate, spice, or even faint florals. Together, they create one of the most complex everyday aromas on Earth. Rich, layered, and immediately readable by the brain before a single sip is taken. Roasting level changes the whole performance. A lighter roast may preserve bright and delicate notes, while a darker roast pushes the chemistry toward bitterness, depth, and smoke. What makes this so remarkable is the sheer degree of transformation involved. The raw seed contains only the potential. The smell that fills a kitchen at dawn is really the sound of chemistry being unlocked by fire. Almond flavor often suggests comfort, sweetness, and dessert. Yet one of its signature notes shares chemical territory with something far darker. Bitter almonds contain compounds that can break down to release hydrogen cyanide, a substance feared for very good reason. Sweet almonds, the kind most people eat, have been carefully bred to avoid that danger and can be enjoyed safely. But the deeper story remains. Plants in this family evolved a way to pack their seeds with defensive chemistry that becomes threatening only when tissues are crushed and enzymes begin to act. The familiar marzipan-like aroma associated with almond therefore carries an unsettling echo of toxicity beneath its sweetness. It is a reminder that pleasant smell and genuine danger do not always live far apart. Nature often hides warning and attraction in the same place, and few flavors reveal that duality so elegantly. Something associated with cakes and pastries turns out to belong to a much older chemical strategy built for defense rather than delight. Willow bark helped inspire one of the world's most familiar medicines. For centuries, people noticed that willow bark could ease pain and reduce fever, and that old observation eventually helped guide one of the most celebrated stories in modern medicine. Willow contains salicin, a compound the body can convert into related forms with pain-relieving effects. Over time, chemists studied that natural chemistry and searched for ways to preserve the benefits while reducing the harshness that older preparations could cause. The result was not a simple copy of the bark itself, but an inspired descendant of it, refined through science into something far more consistent and widely trusted. What makes this story so compelling is the bridge it reveals between folk knowledge and formal medicine. A tree growing beside watercourses and riverbanks became part of a chain of insight that eventually reached the pharmacy shelf. It is a reminder that many breakthroughs do not begin with steel and glass. They begin with bark, bitterness, careful attention, and time. Turmeric stains fingertips, cutting boards, and cooking spoons with a yellow so intense it seems almost to glow from within. The compound behind that famous color is curcumin, a plant pigment that has fascinated cooks, healers, chemists, and medical researchers alike for generations. It helped make curries radiant, ceremonial robes luminous, and food instantly recognizable across entire culinary traditions. Yet the story goes beyond color. Curcumin became one of the most closely examined plant compounds in modern research as scientists worked to understand its behavior in the body and its possible biological effects. That curiosity transformed a kitchen spice into a laboratory regular. Even so, turmeric's most immediate wonder may still be sensory. A dry, knotted root holds a color powerful enough to transform an entire dish with nothing more than a small spoonful. One underground stem stores a pigment that feels like concentrated sunlight drawn up from the soil and fixed into spice. Saffron is one of the clearest proofs that a plant extract can be lavishly complex. Its deep golden color, its hay-like sweetness, and its almost haunting perfume do not come from a single master ingredient. They come from three distinct molecules working in concert. Crocin brings the vivid color that spreads through rice, broth, and cream like liquid gold. Picrocrocin contributes the distinctive bitter edge that keeps saffron from tasting merely sweet. Saffronel rises into the air as the aroma most people recognize, warm, delicate, and nearly impossible to compare with anything else. All of this is concentrated in the dried stigmas of a crocus flower, each one tiny and fragile, gathered painstakingly by hand. That is part of the wonder. Such a grand sensory effect begins in thread-like structures so delicate they must be collected one by one. Saffron does not overwhelm by force. It reveals itself in layers, showing how one small flower can divide beauty among color, taste, and scent with astonishing precision. Clove scent suggests comfort at first. It is warm, rich, and almost festive, the kind of aroma that seems to belong in baked fruit, spiced drinks, and winter kitchens. Yet the same bud contains eugenol, a powerful compound with a very different talent. It can numb tissue and dull pain, which is why clove oil has long been associated with aching teeth and sore gums across many centuries and many cultures. Few spices reveal such a striking contrast between fragrance and function. Something that smells deeply welcoming can also act with real force on the body. That dual nature helps explain the long fascination with cloves in trade, medicine, and cooking. They are not merely flavor. They are concentrated chemistry wrapped inside a dried flower bud. When you taste or smell clove, you are encountering a plant substance that can comfort the senses and influence pain at the same time. One of the clearest reminders that spice and medicine have always, quietly, overlapped. Eucalyptus leaves release invisible vapors that quietly transform the air around them. Stand beneath a eucalyptus tree on a warm afternoon and the atmosphere can feel different, cooler, sharper, almost medicinal. It is not imagination. The tree releases volatile aromatic compounds into the surrounding air, especially molecules such as cineol that evaporate easily and drift outward. For the tree, These compounds may help discourage herbivores, limit microbial growth, and cope with harsh, dry conditions. For us, the experience feels strangely physical, as though the air itself has changed rather than simply carrying a scent. Entire groves can create this effect. As temperatures rise, more of these compounds escape into the breeze and spread across the landscape. Eucalyptus becomes more than something you look at. It becomes something you enter. Its presence extends beyond bark and branches and reaches into the surrounding space. Unlike many plant chemicals that remain hidden inside leaves or stems, eucalyptus projects part of its identity outward into the world. Some plants follow a different strategy entirely. They store important ingredients separately inside their tissues, keeping them inactive until something disturbs them. As long as the leaf remains whole, the chemistry stays quiet. But the moment an insect bites, a hoof tears, or human fingers crush the surface, barriers break apart. Enzymes meet stored compounds, and within seconds entirely new chemistry begins to appear. This is one of the most elegant survival strategies in the plant world. Instead of spending energy producing defensive compounds continuously, plants wait until the exact moment of attack. A sudden bitterness, a burst of scent, or a rapid chemical response can all emerge only when needed. Injury becomes more than damage. It becomes a signal that activates defense. Garlic offers one of the clearest examples of this hidden transformation. A whole clove appears mild and quiet, yet much of its famous intensity remains dormant until its structure is broken. Inside the intact cells, key ingredients stay separated. Slice, crush, or grate the clove and those ingredients finally meet. Enzymes begin working immediately, creating sulfur compounds including allicin, which gives fresh garlic much of its powerful aroma and distinctive bite. That is why crushed garlic smells sharper than untouched garlic. Its identity unfolds only after disruption. For the plant, this chemistry likely evolved as protection against microbes and hungry attackers. For humans, it became one of the most remarkable transformations in cooking. A pale, simple clove can suddenly fill an entire room with scent and character. Few ingredients show more clearly that a plant's true nature may remain hidden until the moment it is broken. Freshly cut grass tells a similar story. That familiar scent drifting across a newly mowed lawn feels calm, bright, and strangely comforting. Yet to the grass itself, it may represent distress. When blades are cut, damaged cells release airborne compounds known as green leaf volatiles. To us, they smell fresh and peaceful. To plants, they may function as alarms. These chemicals can help prepare nearby tissues for defense or attract predators that attack the insects causing the damage. What feels soothing to humans may actually be part of a rapid emergency signal moving through the landscape. That reversal makes plant chemistry unexpectedly fascinating. Human comfort can emerge from signals of survival. The lawn smells alive because the plants have reacted. Invisible messages move through the air from blade to blade, turning what seems peaceful into something active and urgent. Pine forests reveal another version of the same idea. Walk through a pine forest and the air can feel clean and restorative. That familiar fragrance comes largely from terpenes released by needles, bark, and resin. These compounds help defend trees against insects, pathogens, and environmental stress. Some are released gradually in warm weather. Others surge outward after damage as sticky resin seals an injury. To us, the forest feels invigorating. To the tree, it is protection. Entire woodlands breathe chemistry into the atmosphere every day. With every step through a pine forest, you move through an invisible world of signals and survival. The freshness in the air is not passive. It is part of the forest quietly protecting itself, shaping the environment through chemistry as much as through roots, branches, and wood. What tastes like abundance to us often began as engineering. Plants cannot walk their offspring into new places, so many evolved a different solution. They turned sweetness into transportation. Fruits became invitations. Sugars, aromas, and vivid colors worked together to recruit hungry animals into carrying seeds farther than roots could ever reach. What feels generous to us was often designed as a dispersal system, an elegant partnership built over millions of years. Peppermint and spearmint reveal how tiny chemical differences can reshape an entire experience. They are closely related and can seem like two accents of the same flavor. Yet a subtle shift in chemistry sends them in remarkably different directions. Peppermint is dominated by menthol, the compound responsible for that intense cooling sensation associated with strong gum, toothpaste, and medicinal freshness. Spearmint relies more heavily on carvone, which feels softer, sweeter, and more herbal, with far less icy intensity. On paper, the difference may seem small. In the mouth, it feels dramatic. One races across the senses like a cold breeze. The other lingers more gently, greener and rounder, making it especially suited to cooking and confectionery. This is one of the wonders of plant chemistry. Close botanical relationships do not guarantee identical sensory worlds. A subtle change in which molecules are emphasized can transform a familiar leaf from sharp and invigorating into smooth and welcoming, as though neighboring plants had developed entirely different ways of introducing themselves. Basil tells a similar story. Basil is not one single scent wearing different labels. It is more like a whole family of aromatic personalities gathered under one name. Some varieties lean toward warm clove-like notes because they contain higher levels of eugenol. Others shine with bright citrus qualities created by compounds that feel lively and sparkling to the senses. Still others drift toward anise-like sweetness that seems equally at home in gardens and pastry kitchens. This variety is what makes basil endlessly interesting to growers, cooks, and perfumers. The leaves themselves may look only slightly different, yet their aromas can travel in surprisingly different directions. A handful torn into one salad may smell peppery and floral, while another variety feels soft, sweet, and almost lemony. Basil becomes more than a herb. It becomes a reminder that plant chemistry can create multiple sensory identities within a single species, each shaped by genetics, climate, and generations of selection. Citrus peel offers one of the most visible examples of hidden plant chemistry. The skin of an orange or lemon appears smooth from a distance, yet under the surface are thousands of microscopic reservoirs filled with aromatic oil. Twist the peel and those tiny pockets burst instantly, releasing a fragrant mist into the air. It is one of the clearest demonstrations of plant chemistry that can happen in an ordinary kitchen. The aroma is not buried deep inside the fruit. It waits near the surface under gentle pressure, stored in countless miniature chambers. For the plant, these oils likely serve as protection against pests and disease. For us, they become perfume, flavor, and spectacle. A strip of peel expressed over a drink can transform the entire experience before the first sip even reaches the lips. Few ingredients reveal themselves so dramatically. One small twist, and the fruit responds with a cloud of concentrated sunlight and scent. Lemon feels bright not because brightness has a smell, but because its chemistry creates that impression. Much of that effect comes from limonene, a compound abundant in citrus peel that delivers the clean, sparkling quality people associate with freshness and energy. It is not sweet in the way vanilla feels sweet, and it is not floral like rose. It feels sharper, faster, almost visual. That is why lemon appears so often in kitchens, perfumes, cleaning products, and candies that want to suggest clarity and renewal. Yet beneath that cheerful impression lies another purpose. Limonene exists because the fruit evolved a protective chemical barrier across its surface, so when a lemon is peeled or zest, what escapes is not simply fragrance. It is a defensive chemistry that humans learn to associate with light, cleanliness, and awakening. The orange tree reveals another layer of this story. It seems natural to assume one tree should have one signature scent, but orange trees speak in more than one language. Their blossoms release a soft fragrance that feels airy, creamy, and delicate. Their fruit peels release sharp citrus oils that feel vivid and immediate. Same tree. Entirely different aromatic worlds. That contrast exists because flowers and fruits are solving different problems. The blossom must attract pollinators with precision. The peel must defend developing fruit with a stronger chemical shield. One part of the tree whispers. Another part flashes. Perfumers have treasured this distinction for centuries, capturing narrowly and orange flower water from blossoms while cooks and distillers turn to zest and peel for brightness. The orange tree becomes a living lesson in plant chemistry, showing how one organism can speak in multiple scents at once. Grapes continue the story in an even quieter way. A fresh grape can taste simple, juicy, and sweet. Yet hidden inside are chemical possibilities that may not fully appear until yeast enters the story. During fermentation, sugars fade, alcohol emerges, acids shift, and an entirely new world of aroma slowly begins to unfold. Some aromas remain hidden until transformation begins. Inside fresh grapes, certain compounds exist in bound forms that carry little scent on their own. Others remain dormant until yeast enters the process and begins reshaping them. During fermentation, these hidden ingredients are released or transformed into entirely new aromatic worlds. Notes that can suggest flowers, stone fruit, spice, herbs, smoke, or earth. That is why wine can feel far more intricate than the grapes from which it came. Fermentation is not simply preservation. It is revelation. The fruit arrives carrying locked doors, and microbial life begins opening them one by one. What finally emerges depends on countless details, including grape variety, climate, skin contact, timing, and even the strains of yeast involved. A vineyard is not only growing sweetness for harvest. It is quietly cultivating ingredients for aromas that may not fully exist until the fruit becomes something new. Hops tell another story of transformation. The bitterness in beer can feel refined, refreshing, and deeply satisfying. Yet the compounds behind that experience evolved for a very different purpose. Hop plants produce bitter acids and aromatic resins as protection against pests and microbial threats. Humans discovered that these same defensive compounds could transform brewing. They balanced sweetness for malt, added structure to flavor, and helped beer remain stable for longer periods. That combination changed brewing history. Hops did not simply season beer, they expanded what beer could become. Their chemistry opened entirely new possibilities of aroma and expression, ranging from pine-like and resinous to floral, citrusy, and tropical depending on variety and handling. Every crisp finish, therefore, carries an ecological backstory. What feels refreshing in a glass began as part of a plant's survival toolkit before becoming one of humanity's most enduring fermentation traditions. Cinnamon reveals how precise plant chemistry can become. Its warmth feels almost designed for memory. It settles into pastries, warm drinks, and winter kitchens with a softness that seems to glow. Yet cinnamon's famous character does not come from the wood of the tree itself. It comes from the inner bark. That bark is harvested, dried, and curled into familiar quills as aromatic compounds become concentrated. Much of cinnamon's recognizable identity comes from cinnamaldehyde, supported by many smaller molecules that add depth and complexity. This reminds us that plants are not chemically uniform. Different layers tell different stories. Within the same trunk, one layer becomes structure while another becomes fragrance. The bark protects living tissues beneath while carrying chemistry that humans have treasured across centuries of trade and culture. A cinnamon stick is therefore not carved wood. It is the fragrant skin of a tree. Black pepper creates a completely different experience. It does not burn like chili and it does not cool like mint. Instead, it spreads slowly with a dry, warm bite that deepens as it lingers. That sensation comes mainly from piperine, a compound notable not only for flavor but for remarkable stability. Unlike delicate aromas that disappear quickly with heat or time, piperine remains persistent through grinding, transport, and cooking. That durability helps explain why pepper became one of history's great traded spices. Tiny dark berries from a climbing vine could travel enormous distances and still deliver their unmistakable spark. The plant likely evolved that chemistry as defense, but humans interpreted something else entirely. We tasted warmth, energy, and depth. In one small peppercorn, protective chemistry became one of the world's most universal seasonings. Cardamom carries another lesson in concentration. Few spices feel so disproportionate to their size. Crack open the pod and inside are tiny dark seeds carrying fragrance powerful enough to reshape an entire pot of tea, a dish of rice, or a tray of pastries. Its aroma resists simple description because it seems to move in multiple directions at once. At different moments, it can feel citrusy, cool, resinous, sweet, and warmly spiced. That complexity comes from a dense collection of volatile compounds stored inside minute amounts of seed tissue. The scale of it feels almost impossible. Something smaller than a fingernail can perfume an entire room. That efficiency is one reason cardamom became treasured across cuisines and trade routes spanning continents. It offers intensity without heaviness and brightness without harshness. The pod appears modest, but once opened, it releases something far larger than the structure that contained it. Coriander offers one final surprise. Few culinary plants create such contrast within themselves. Its fresh leaves can feel vivid, green, and exhilarating to some people, while others experience them as soapy or sharp because of differences in scent perception. The seeds tell a completely different story. Once dried, toasted, or ground, they become warmer, softer, nuttier, and touched with gentle citrus sweetness. Same plant, entirely different expression. It is a reminder that even within one species, plant chemistry can create sensory worlds that barely seem related. This happens because the chemistry of leafy tissues is not the same as the chemistry of mature fruiting structures. Different compounds dominate at different stages and in different parts of the plant. As a result, the plant changes its sensory language throughout its life. That makes coriander more than a seasoning. It becomes a small lesson in botanical complexity. A single species can offer two nearly separate flavor identities, one vivid and divisive, the other softer and more widely loved, without ever changing its name. Fennel tells a different story. Its aroma is recognizable long before many people can name it. Sweet, herbal, and gently cool, it carries notes that drift toward old herbal sweets, traditional remedies, and apothecary shelves. Much of this character comes from anethole, a compound powerful enough to define fennel's identity while appearing in a small group of other plants known for similar flavors. What makes fennel fascinating is the way it sits between comfort and intensity. In one setting it feels culinary, in another medicinal, and in another almost perfumed. That flexibility has allowed fennel to move naturally between kitchens and traditional remedies for centuries. Seeds, fronds, and bulbs all carry versions of the same identity, although each expresses it with different balances and textures. When crushed or extracted, the sweetness appears with remarkable clarity. It becomes one of those plant signatures that seems almost attached to memory itself. A small trace can instantly call to mind bread, herbal tea, sweets, or medicine. Star anise reveals how chemistry can connect plants that seem unrelated. At first glance, star anise and fennel appear completely different. One arrives as a dark, star-shaped fruit with dramatic structure. The other appears as delicate fronds, seeds, or a pale garden bulb. Yet the moment their aromas rise, the relationship becomes obvious. Both contain large amounts of anethole, the molecule responsible for their familiar licorice-like sweetness. This shared chemistry shows that plants do not always need close ancestry to create similar sensory experiences. Appearance and culinary role may differ completely while the underlying molecular language remains familiar. That is why star anise can echo fennel in broths. Teas, sweets, and spice blends while still bringing its own deeper and more resonant character. Beneath the surface, both plants are speaking variations of the same aromatic language. It is a reminder that flavor is not always shaped first by appearance or origin. Sometimes chemistry creates the family resemblance. Rosemary demonstrates that fragrance can serve hidden purposes. It is easy to admire rosemary for its pine-like and savory aroma, but one of its most practical qualities is less visible. Compounds inside the plant can help protect fats from oxidation, the process that slowly pushes oils and rich foods toward stale or rancid flavors. In other words, rosemary does not simply season food. It can also help preserve it. That makes sense in the life of the plant itself. A shrub growing under intense sunlight and dry conditions benefits from chemistry designed to manage stress and protect living tissues. Humans learned to borrow that advantage. Rosemary extracts have been used to help stabilize foods rich in oils without depending entirely on synthetic additives. It is another example of a flavoring herb quietly doing important work behind the scenes. The scent rising from a roasting pan may be the most obvious contribution, but another part of the plant's chemistry is silently defending freshness. A fragrant needle leaf becomes a small guardian against time and air. Thyme carries a scent that feels unusually clean, almost as though brightness and seriousness exist together in the same aroma. Much of that character reflects compounds such as thymol, which help the plant defend itself against microbes and environmental pressure in the dry landscapes where thyme often grows. Its fragrance is not decorative. It is active chemistry with protective intent. Humans recognized that quality long ago. Thyme found a place not only in cooking, but also in traditions of preservation, cleansing, and herbal practice because its scent suggested strength beneath the surface. Even a small pinch can dominate a dish, cutting through richness with remarkable precision. That intensity comes from a plant that survives exposure to heat, dryness, and constant challenge by concentrating protection into its leaves. When thyme rises from a hot pan or crushed sprig, what reaches the air is a Mediterranean survival system translated into flavor. Sage continues the pattern. Run your fingers across a sage leaf and it already feels different from many herbs, soft, textured, almost velvety, as though designed to endure difficult weather. Inside those leaves is equally purposeful chemistry. Sage stores rich aromatic oils that help it withstand strong sunlight, dry conditions, insects, and microbial threats. For us, those compounds become a fragrance that feels earthy, broad, and slightly smoky with enough strength to anchor butter, stuffing, tea, and countless other dishes. Behind that culinary depth lies a survival story. Plants growing in demanding climates often invest heavily in protective chemistry, and sage is an elegant example of that strategy. Its leaves are not flavorful by accident. They are concentrated with compounds because the plant must endure. What finally reaches the kitchen becomes a memory of sun, stress, and resilience. Oregano offers one of the strongest examples of defensive chemistry becoming flavor. It can seem oversized for an herb. A small pinch can fill a sauce, rise through steam, and linger long after the heat disappears. That force comes from compounds such as carvacrol and others that help protect the plant against microbes, grazing animals, and environmental stress. In nature, that intensity supports survival. In cooking, it becomes one of the most recognizable herb flavors in the world. Oregano does not arrive quietly in a dish. It announces itself immediately. Even after drying, it keeps remarkable strength because the chemistry underneath remains stable and resilient. What feels bold in the kitchen began as protection in the wild. That strength made oregano especially valuable in cuisines built around herbs that could survive storage, endure sunlight, and continue delivering flavor through long cooking. What tastes bold in tomato sauces, roasted vegetables, and slow-cooked dishes began as concentrated botanical defense adapted beautifully into flavor. Bay leaves reveal a different kind of power. A single leaf often looks too modest to matter. Slip one into a pot, and at first it seems to do almost nothing at all. But heat and time slowly change that impression. As the leaf simmers, aromatic compounds begin to loosen and move into the surrounding liquid, releasing flavor that feels subtle yet quietly structural. Bay does not announce itself the way oregano does or arrive instantly like citrus peel. It behaves more like an infusion, gradually building depth, dryness, and a certain herbal lift that can be difficult to identify until it is absent. That slow release is part of its character. The leaf evolved to hold its chemistry tightly, helping the plant endure changing conditions. Cooking simply persuades those compounds to emerge. What appears to be a dry, rigid leaf becomes, with patience and warmth, an architect of flavor moving silently through an entire dish. Vanilla tells an even more surprising story. Freshly harvested vanilla pods often disappoint expectations because they do not immediately smell like vanilla. The familiar aroma most people recognize is not waiting there fully formed. It appears through curing, a carefully controlled process of sweating, drying, resting, and gradual transformation. During this period, enzymes and stored compounds inside the pod begin changing, allowing vanillin and many supporting aromas to emerge. This means vanilla is not simply harvested. It is developed. Human skill becomes part of the final flavor, guiding the pod through a slow journey from green and quiet to dark, fragrant, and deeply complex. That transformation helps explain why vanilla became so prized throughout history. The the sweetness is not effortless. It arrives through patience, labor, and careful timing. Behind every familiar soft note is an orchid fruit that had to be persuaded over weeks and months to reveal the scent now recognized around the world. Cocoa follows a similar path of transformation. Fresh cocoa beans taste nothing like the smooth chocolate most people imagine. Straight from the pod, they are bitter, sharp, and surrounded by sweet pulp with very little of the rich aroma that later defines chocolate. The transformation begins after harvest when the beans ferment beneath leaves or inside boxes, warmed by microbes feeding on surrounding sugars. This process changes the chemistry inside the seed, reducing harshness and creating the foundations of future flavor. Then come drying, roasting, and grinding. Heat drives further reactions that build the nutty, roasted, and complex notes people instantly recognize. Chocolate is therefore not discovered, it is developed. A tropical seed packed with defensive bitterness becomes one of the world's great pleasures only after microbes, time, and heat complete their work. Few plant products show transformation more dramatically. The bean begins almost unrecognizable and slowly becomes something millions of people crave. Tea offers another kind of chemistry entirely. The dry, gripping sensation strong tea can leave behind is not dehydration. It is chemistry unfolding directly on the surfaces of the mouth. Tea contains tannins, compounds that bind to proteins in saliva. As this happens, the mouth loses some of its smoothness and the tongue begins to feel tighter, rougher, and drier. The effect can be delicate in lightly brewed tea or strong and unmistakable in darker infusions. That sensation helps explain why tea feels structured. It is not only aroma and taste. It has texture, and that texture emerges through direct interaction between plant compounds and the body. It also explains why milk, sugar, or shorter brewing can change the experience so dramatically. They soften the grip. What seems at first like a simple drink is quietly creating a tactile event that lingers long after the sip itself disappears. Oak bark reveals how plant chemistry shaped human history. For centuries, oak was more than timber and shade. Its bark contained tannins powerful enough to transform animal hides into leather, converting something temporary into material strong enough for shoes, saddles, belts, and books. The process worked because tannins bind to proteins, strengthening and stabilizing the hide in ways that resist decay. A defensive chemistry evolved by trees became central to one of civilization's most important crafts. Tanneries once relied on bark that was stripped, ground, soaked, and slowly released into hides over time. The work was demanding and often difficult, yet it connected forest chemistry directly to everyday life. A tree growing quietly at the edge of a woodland could eventually shape clothing, travel, and trade. Few examples show more clearly how plant extracts moved from ecology into material culture. Pomegranate shows how differently chemistry can be distributed inside a single fruit. The bright red seeds feel lush and jewel-like, yet the rind and inner membranes tell a tougher story. These outer layers contain far more astringent compounds than the sweet tart portions people usually eat. Bite into those pale sections by accident and the mouth can suddenly feel gripped and dry. That contrast reveals a careful design. The edible portions attract while the outer structures defend. The fruit presents sweetness protected behind chemistry built to discourage unnecessary attention. Traditional uses valued this astringency not as food but as something useful in extraction and preparation. The pomegranate becomes more than a container of sweetness. It resembles a fortress, a brilliant center protected by layers designed to keep the world at a distance. Desert plants carry this idea even further. In deserts, danger does not only crawl, bite, or infect. It also arrives from above. Intense sunlight can damage tissues, disrupt essential chemistry, and push living cells toward breakdown. Some desert plants answer with protective compounds powerful enough to defend not only against animals and microbes, but against the pressure of light itself. Some desert plants respond to extreme environments by building powerful protective compounds directly into their skins, waxes, and outer tissues, creating something like a chemical shield against ultraviolet stress and relentless heat. These compounds may absorb harmful radiation, neutralize damaging byproducts, or help stabilize the plant's own delicate systems under constant exposure. The plant is not simply enduring the sun. It is chemically negotiating with it every day. That is why desert leaves are often small, silvery, resinous, or deeply scented, visible signs of a life shaped by defending against brightness itself. To us, a desert shrub may appear motionless and rugged. In reality, it is maintaining an ongoing chemical defense against one of the harshest environments on Earth. Its extracts are not only protection against insects or disease. They are part of a survival strategy written directly against light. Plants also reveal another remarkable strategy: timing. Some of the most sophisticated plant defenses depend on keeping powerful chemistry hidden until the exact moment it is needed. Certain compounds would be dangerous if active inside the plant itself. So many species separate ingredients until an animal bites, tears, or chews the tissue. Only when cells break do enzymes and stored compounds finally meet, creating defensive molecules at the moment of attack. It is an elegant solution. The plant avoids harming itself while keeping an effective deterrent ready for immediate release. For the attacker, the result may be bitterness, burning, irritation, or something stronger, triggered only after damage begins. A leaf or seed becomes something like a chemical puzzle box, quiet on the surface, active only when forced open. In this way, the bite itself becomes the trigger. Injury transforms into retaliation. Mustard offers one of the clearest examples of this strategy. A mustard seed seems small and inactive, yet its real force sits behind a chemical lock. Inside the intact tissue, important ingredients remain separated. Crush the seed and the barriers disappear. Enzymes begin transforming stored compounds into pungent molecules that rush upward with that familiar sharp sting. This is why mustard feels especially alive when freshly ground or mixed. This heat is not stored the way chili stores heat. It is generated in the moment through disruption. The defense appears because damage occurred. That is what makes mustard so fascinating. The seed does not merely contain flavor. It contains the potential for flavor, waiting for moisture, pressure, and rupture to bring it into existence. Something that looked quiet only seconds earlier suddenly becomes intense and vivid, reminding us again that plants often reveal their strongest character only when disturbed. Some plant extracts vanish quickly because they are chemically restless. Certain fragrances bloom brilliantly and disappear almost immediately. Citrus zest brightens the air and fades with surprising speed. Crushed herbs burst into life and then soften before the moment fully settles. This happens because many of the molecules involved are highly volatile. They evaporate rapidly, react with oxygen, or shift under warmth and light. Their fleeting nature is part of their beauty. They feel alive because they refuse to remain still. In plants, these compounds may move quickly to signal, defend, or communicate. In human experience, they become the sparkling top notes of perfume and the first impression of freshness. A scent that disappears quickly is not weak. Often it is simply made of molecules eager to escape and transform. Other scents behave very differently and remain for hours. Not all plant fragrances vanish on the breeze. Some settle into fabric, skin, wood, or hair and continue releasing themselves slowly. These longer-lasting effects usually come from heavier and less volatile compounds. Resins, woods, roots, and deeper floral materials often carry this kind of persistence. Their aromas unfold gradually but continue returning with warmth and movement. This is why a scarf may still smell faintly of perfume the next day, or why incense lingers long after the smoke disappears. These extracts become the memory keepers of fragrance, staying behind after the brighter notes have gone. The future of medicine may still be hidden in overlooked plants. Although many medicines already trace their origins to leaves, bark, roots, and resins, the botanical world remains vastly unexplored. Countless plant species have never been deeply studied, and even known species may contain overlooked chemistry depending on where and how they grow. Plants have spent millions of years evolving molecules that interact with living systems in highly specific ways. They have been solving problems of defense, repair, signaling, and survival far longer than human laboratories have existed. Somewhere inside that enormous living library may be future treatments for infection, pain, cancer, or challenges not yet understood. Discovery does not always begin in advanced technology. Sometimes it begins by paying attention to something local, unusual, or ignored. The next major breakthrough may still be growing quietly in plain sight. Plant extracts ultimately reveal that rooted life is far more active than it first appears. Plants may seem passive because they do not run, hunt, or speak, but their chemistry tells another story. They create coolness without cold, heat without fire, fragrances that recruit insects, pigments that change with conditions, and compounds that become medicines, dyes, preservatives, stimulants, and defenses. Every root, seed, bark, stem, petal, and leaf may hold the memory of some ancient challenge expressed not through movement but through molecules. Plants do not stand still. Their lives unfold through chemistry. Some molecules create the instantly recognizable note in a plant's scent. Others work more quietly, rounding the edges, sharpening brightness, adding sweetness, or creating subtle shadows that most people only notice when they disappear. This is why natural aromas feel so alive. They are never one ingredient pretending to be complete. They are entire chemical communities moving together. Take lavender, for example. Its fragrance may seem simple and familiar at first breath, but its identity depends on dozens, sometimes hundreds, of compounds arriving in careful balance. Shift that balance enough and something changes. The scent becomes flatter, less vivid, less alive. What feels effortless to the senses is often breathtakingly complex at the molecular level. The plant offers an orchestra, and the mind hears a single melody. Even more remarkable, tiny changes in molecular shape can transform aroma completely. Some molecules contain the exact same atoms connected in the exact same order, yet exist as mirror images of one another. This phenomenon is called chirality, and in the world of scent it can change everything. One version may smell bright and citrus-like, while its mirror image feels closer to pine or resin. On paper, they appear nearly identical. To the nose, they are entirely different experiences. That happens because smell itself is three-dimensional. Our scent receptors respond not only to ingredients but also to how those ingredients are arranged in space. A slight twist in structure becomes a completely different sensory experience. Plant extracts, therefore, carry more than chemistry alone. They carry geometry. Nature works in dimensions our senses quietly detect without us ever needing to think about it. Some plant scents disappear almost as quickly as they arrive. Citrus peels brighten the air and then fade. Fresh herbs burst into life and soften moments later. These fragrances are built from highly volatile compounds, molecules eager to evaporate, react, and drift away. Their fleeting nature is not weakness. It is part of what makes them feel vivid and alive. Inside the plant, those fast-moving compounds may serve important purposes: signaling, attracting, warning, or defending. In human experience, they become the sparkling top notes of perfume and the freshness we associate with crushed leaves and ripe fruit. Their beauty comes partly from their refusal to stay. Other aromas behave differently. They settle into fabric, wood, hair, and skin, unfolding slowly over hours. These longer-lasting impressions often come from heavier molecules found in resins, roots, woods, and deep floral extracts. They do not rush into the air. Instead, they linger, returning softly as warmth and movement release them again. That is why a scarf can still carry perfume the next morning, or why incense leaves traces long after the smoke disappears. Some plant chemistry is built not for arrival, but for staying. These lingering notes become the memory keepers of scent, the quiet echoes left behind after the brighter notes have already flown, and perhaps that lingering quality mirrors the larger story of plants themselves. Their world is patient. Rooted and silent, they build complexity molecule by molecule, season by season, until entire hidden worlds are folded into a single flower, a strip of bark, or one unnoticed leaf. Once you begin to notice it, stillness no longer feels empty, feels full of invention.